So let's get it started. We are just moving slowly ahead as a small snail here on the right hand side, but I do think that we now have an option to develop IVM, human IVM, in a way where we have never ever had it before. And I really do think that there is a future for doing human IVM. So this is what I would like to focus on. I actually want to tell you that in our hands, we find a surprisingly high number of immature oocytes in the surplus medulla tissue. And it do come out that these oocytes have different quality. And we now start to know, we start to learn how these oocytes, which we find in the medulla tissue, how are they actually reacting to one when we try to do IVM, and what is the important parameters to control for. And then we have identified a new interesting growth factor, midkine, which appears to be very important for the acquisition of the ability to go into metaphase 2 of these oocytes. I would like to discuss this with you. And then obviously these oocytes are from small antral follicles. Would aneuploidy be increased? Are they as good as those that we normally collect? Let's discuss this. And then of course, I would like to dwell a little bit on the perspectives. And I can already now tell you that if we start to consider IVM, there's actually quite a lot of more patients who can benefit from ovarian tissue freezing. So as you are very well aware, this is just the cross-section of an ovary, and all the small white dots out here, this is what we collect for ovarian tissue freezing. This is the primordial follicles. These are the only ones which sustain freezing, and we do not consider all the medulla tissue and all the growing follicles. So is there any opportunity for us to look into this part of the tissue to collect any fertility preservation potential? Obviously, the ovarian tissue freezing is favored by the fact that the number of follicles here in the cortex is always more than 90% of whatever follicles is available in an ovary at any moment in time. But there is a lot of antral follicles as well. Just to keep everybody on track, this is the way we do it. Collect one ovary, prepare the cortical tissue. We put them into pieces like this. I just watched the previous presentation. We would do pieces which would be 5, 6, 7 millimeter in this, and then around the same length in this size, and then obviously the thickness. The 1 millimeter thickness is the important part. Freeze it, saw it out, and then transplant it, in this case, to the remaining postmenopausal ovary. So this is the cortical part. This is not what we're going to deal with today. We're going to look into the medulla part. So this is how it would look in our lab. When we had one ovary, the cortical tissue has been prepared here, 2 halves. This has been prepared. We will cut this into small pieces and then freeze it. This is the rest of the ovary. This is what is left over from this part. So at one point I said to all the people working, so have a look as if this was an IVF procedure. Would you be able to locate any immature oocytes, not follicles, but immature oocytes released from antral follicles? Obviously, we would cut this up. And if we do look into this, it do come out that there is surprisingly high number of immature oocytes, which are released from small antral follicles with a diameter of, say, maybe 0.5 millimeter to 1 to 3 millimeters. And they do come out very differently. You see here, huge, this is what we call large cumulus oocyte complexes. Here's the oocyte. There's some which are small, and there's some which are almost naked. And this one is almost naked. This actually appears or comes out as a very important quality measure of these oocytes. So, but then when you start to look for them, suddenly you realize that what you have now normally just been throwing in the rubbish bin actually contain valuable material. And if you look at it, then IVM is this is the way everybody else, except for humans, is doing IVF. You have almost any species you can think of. This is sheep, goat, bovine, pig, whatever. You collect one ovary, you aspirate the the antral follicles, and then you mature them in vitro, and you have beautiful offspring. Everything is good and normal, and this is basically the standard method. But why doesn't this work in humans? Well, up until now, we have never ever had one ovary where we could aspirate the oocytes from. We are always just aspirating small antral follicles, and maybe there is actually something in the size of the follicles which I would like to return to. So what we're looking at is these. This is half a human ovary. These are the small antral follicles. Obviously, those we can aspirate directly with a small syringe, and then there will be a lot of small antral follicles which we basically don't see with the naked eye. So this is what we're looking at. And I'm sure that a lot of you would be sitting out there and saying, okay, so he wants to collect oocytes from follicles that is around 1 to 3 millimeter. Come on. We know that if we do IVM, we have tried that. Many people have tried that. 3 days of FSH stimulation and HEG trigger, and you collect oocytes from follicles 10 to 14 millimeter in diameter. Well, you can get pregnancies out of it, but it doesn't work as good as IVF, and generally it is abandoned. I would actually like to raise the possibility that perhaps the oocytes that we can collect from the small follicles are even better. So what I want to talk to you about now is that we are now moving down the ladder of looking into what human follicles, how they grow and how they're regulated. Of course, we are normally just considering those which are just about to ovulate in connection with IVF. If we do normal IVM, this is what we're looking at. But now we're all the way down here. So this is much earlier. And why would we ever consider to look into them? You would think that it would be a much more difficult task to use these compared to those up here. But we're now slowly moving down here. Let's just have a look. Actually, it do turn out that these small follicles have a much higher FSH receptor expression as compared to larger follicles. So we may have a better opportunity to actually stimulate them with FSH in vitro, because FSH receptor expression do become very significantly downregulated as the follicle advances to the preovulatory stage. This is also seen in this picture here, where we have the follicular diameter down here. We have the FSH receptor expression here, and you can see that it steeply decreases when we advance from 2, 3, 4 millimeters and then to larger follicles. In vivo, this is of course reflected by the fact that at this point, LH receptors start to be expressed, and they drive the follicle. But we can actually drive the follicle in vitro by FSH. Why is this interesting? Let's have a look at this wheel from Alain Duchamp, the very classical way of looking at follicular genesis all the way in humans. You can see that going from here 150 days, this is almost half a year going all the way around. What we normally do in IVF.
Professor Claus, I'm sorry. Is this your maximum volume? Can we increase the volume?
Can I increase the volume?
You were perfect when you began. Do you want to try using the headphones again? Or
I can try to do that.
Have enough time for your session, so we can hear you again a couple of slides back too. We have enough time for you. Can you speak?
Where do I have a...
Perfect.
Is this better?
Yeah, this is perfect. Can we go back a few slides if possible?
Okay. All right. Okay. I'm really sorry for this. Okay. So what I was alluding to a little earlier was the fact that the small follicles actually have a very high expression of FSH receptors. So you can see here that in small follicles, the FSH receptor expression is significantly and much higher than in the preovulatory stages, which means that we may have a better opportunity to stimulate the oocyte humus complexes in vitro, because there's a lot more FSH receptors there. And if you see this slide, you can see that this is the follicular diameter down here, and you can see that FSH receptor expression is very high and coming to a plateau when they follicles reach a diameter of 6 to 7 millimeters. This is where LH receptor starts to be expressed, and they drive folliculogenesis from there on. But if we look at cumulus oocyte complexes down here, we may have an opportunity to stimulate with FSH, which I think is very interesting. And as I said, this is the follicular wheel by Alain Bouchon, the classical, which he published now 25 years ago. And here you see that the development of human follicles takes half a year. What we normally consider in context of IVF is that we start to stimulate follicles when they are less than 2, 3, 4, 5 millimeters. The interesting thing is that Alain Bouchon actually looked at the atresia rate of follicles at these stages. And look here, 77%, 58% of the follicles are actually atretic during normal circumstances. This is because that they have been experiencing too little FSH stimulation in the early part of the follicular phase. But if you look at these follicles, they actually have very much reduced atresia rate. So if we start to consider these cumulus oocyte complexes, the atresia is a lot lower, and maybe we can stimulate them and then get them past this selection step, which happens in vivo. So if we stimulate them here, we may actually take them up there, up to the mature state in a fast, accelerated step, and then avoid the atresia steps or the atresia mechanisms that kick in during normal follicular development. So it may be interesting also from a theoretical point of view. So I think that this actually allows us to study human ovaries for the first time in a way we have never done before. And I think I'm absolutely sure that we can develop new strategies for assisted reproduction altogether by starting to study the earlier stages of follicular development. So I will just highlight before I continue that we have in some patients who have PCOS been able to collect more than 100 oocytes from one ovary. I think we have collected 132 oocytes from one ovary, which of course is a massive amount of oocytes which is suddenly available and which we normally just throw out. You can see here large oocyte cumulus complexes, and here are almost naked. So of course there is a huge variety, and this is what we would expect. Some are healthy and some are atretic. After a 44-hour IVM culture, this is the degenerated, the germinal vesicle here, the metaphase 1 and the metaphase 2. So how many metaphase 2 oocytes? This looks very nice. How many do we actually get? Well, here we did a study which came out in JARC this year. 25 consecutive patients who allowed us to look for IVM oocytes, 28 years of age. The ovarian volume was 9, so they had normal ovaries. Obviously, those are the PCO ovaries. But the interesting thing, and I do acknowledge that we had 2 PCO women in this study, but on average, we collected 36 oocytes from one ovary. So this comes as an additional surplus to us. And we had around 31% which matured to metaphase 2, but this is 11 metaphase 2 oocytes on average. This is as good as any ovarian stimulation. If you obtain 11 metaphase 2 oocytes from an ovarian stimulation, you're successful. And this comes absolutely for free. And this is why I call it 2 for 1. If we look at how well the different categories of oocytes are doing, naked oocytes don't do very well. Small cumulus oocytes do okay, but the large COCs, those with more than 10 layers of somatic cells surrounding the oocyte, do half of them actually mature. And you can see that they are also having a cumulus expansion, which is pretty high. So the large COCs are very interesting in the context of fertility preservation. If we now start to look at the oocyte itself, it do turn out that the diameter of the oocyte is an important quality measure, because it actually turns out that the higher the diameter is, the better it makes it. Those which are metaphase 2 have significantly higher diameters than those which do not make it. So the diameter of the oocyte at this stage, the zonal pellucidal thickness is not of importance, but the diameter is. And as you may know, we have this transport system in Denmark where we obtain ovaries from different parts of the country. They are transported to us in 4 degrees or on ice, and we are now starting to look at the collection media, whether we just use saline or HEPAs, and then this growth factor, as I talked about, midkind, I'll come back to that. But you can see if we transport it and the ovary is cooled on ice for say 4 or 5 hours, we obviously get a better maturation rate if it's the local hospital where we just collect the fresh ovary and where it's not cooled off. But the interesting part is that despite the fact that the ovary has been cooled off to 0 degrees for say 4 hours, there's still 25% which mature. If we collect the media where we prepare the cortical tissue, use buffered media, we get a higher maturation rate than if we just use saline. And this growth factor midkind actually also significantly advances the frequency by which oocytes enter metaphase 2. So there's a lot of different conditions that we can optimize to enhance the number of metaphase 2 oocytes we have available. And now I would like to just show you the study that we have done in order to localize that midkind as a growth factor of importance. This is done in collaboration with these 3 guys, and they are very proud because they went to, they are part of a cancer moonshot program in which the current president-elect or president who wants to be president in Joe Biden, he's actually the mentor for this, so they did this. So they're very happy to show this picture. But we have looked at the small antral follicle. Say we have a follicle 5 millimeter in diameter. There's a small, tiny fraction of follicular fluid. These guys are very good at doing proteomics. So they have done analysis of what kind of proteins are in this fluid. And as you see here, we have done a study now, which is about to be published in Human Reproduction, where we have now identified more than 2,000 different proteins in this follicular fluid. And you can see this is what was already known in the literature, but 1,100 were new proteins, never been described in follicular fluid before. This is the abundance, and you can see here the rank of abundance here is now the least abundant one, and you can see here polystatin, AMH, inhibin B, inhibin A, and midkind was then localized somewhere down here. It was there, but the interesting thing is that if we look at those oocytes which were exposed to midkind, they had a significantly higher maturation rate. We mentioned, which is very closely linked to midkind, showed the same thing. And if you look, we had some patients where we collected oocytes from antral follicles where one did not mature and the other one did. Or in this case, there were 2 oocytes that matured. And in these paired samples from one woman, one oocyte made it, one oocyte didn't make it to metaphase 2. The concentration of midkind was outstanding as the one which depicted or characterized this oocyte. So this is very interesting. And if we look, this has been used in bovine oocytes. You can see here that the nuclear maturation is not different, but if you look at blastocyst formation on day 7, significantly enhanced, it doubled here, it's doubled here. So there, and it do turn out that midkind has an important promoting effect on metaphase 2 transition in human oocyte. It's the midkind comes from mid-gestation kidney, shortening of that. It's an anti-apoptotic growth factor. It's pretty short, 121 amino acids. It's interacting with heparin glycosamines, glycans, and it do turn out that it's heavily expressed in cumulus cells, and the receptor notch, which is signaled through, is also present in cumulus cells, and this is all the well-known intercellular pathways, the MAP kinase, the PIK kinase pathways, and it apparently has an impact on the way that human oocytes are then undergoing metaphase 2 transition. We have done another study, where we have published a few studies this year in 2002, where we have been able to collect human follicular fluid during the mid-cycle surge of gonadotrophins. So at 0 hours, this is when we gave the HCG injection to the patient. We took out the follicular fluid sample, and then we waited 12 hours, and then another patient had one follicle aspirated, and another group of patients had one aspirated at 17 hours, 32, and 36 hours. So we tried to cover the whole mid-cycle surge of gonadotrophins and started to look at, amphiregulin is an EGF-like peptide, which also has a huge impact on oocyte maturation in humans. And you can see that it goes from hardly anything to a peak at 17 hours. The same does midkind. And this is just around the time when the human oocyte actually undergoes GVBD and reaches metaphase 2 in vivo. And it's very nicely depicts that in vivo, these growth factors are also important for human oocyte maturation. So obviously it's interesting to try and study them in vitro. And it do come out that if we look at this oocyte diameter and say at which diameter of oocytes will we predict that 50% of the oocytes will undergo maturation, you can see that it moves downward when we increase the conditions. So if we use transported ovaries and only use saline, the diameters are very high. If we transport them and use HEBIS, you don't hear me again? You do. Okay, very good. I'll continue. But as you see, if we do no transport and use HEBIS buffer, this is then we are moving down here 50%. The oocyte diameter becomes lower, which means that we can then have more oocytes which actually do what we want them to undergo metaphase 2 transition. And therefore, it shows us that we have an opportunity to make this better. We can make more oocytes into the metaphase 2, which is what we want. In collaboration with Professor Eva Hoffman at the University here in Copenhagen, we have been looking at the aneuploidy rate of oocytes, which was matured to the metaphase 2 state, and actually we come out with a frequency of something like that 2/3 of the oocytes are chromosomal normal after our IVM procedure, which is a little lower than what you see in IVF, where you would have something in the early 70s. So the oocytes are almost as good aneuploidy-wise as those that we would get in IVF, which of course is very interesting. What we do have as a stop clause for us here in Denmark is that the authorities do not recognize IVM as a standard procedure, which means that we cannot do it clinically. And therefore, we have been unable to fertilize the oocytes. We are working on that with the authorities, but this is very difficult, and we probably have to do a lot of clinical studies to be allowed to do it, which of course is very annoying, because I do think that it's already shown out in the big world that it is an option, and we do not do any harm to the oocyte or the outcome of those children that would be a result of it. But I think one important question is to ask. So this large cumulus oocyte complex, where you see the oocyte here, there's a huge somatic cell component here, which we can start to stimulate with FSH, with midkind, and different factors. And obviously they are closely linked to the oocyte, and they can determine if we can make this oocyte mature in vitro faster. So what I do think comes out is that this very low atresia rate here may be a benefit, because we can take these oocytes and stimulate them in vitro, and thereby surpass this step where a lot of atresia takes place, and then accelerate the oocytes to this step. And then maybe we can use them, which I think is a very interesting option. Of course, the first goal is to use fertility preservation and use the cortical tissue, but we do have the additional medulla tissue. So I think it's fair now to say that we can do 2 for 1. We can do almost as good as an IVF ovarian stimulation cycle by doing IVM. And what I do think is the very interesting part, and we have started to do that now, is that for the first time ever, we are now offering women who have ovarian cancer or borderline ovarian cancer, where we would never ever think of transplanting the tissue because we would potentially replace the cancer. But if we can collect oocytes, we know that the oocytes were created when this was a fetus and there's no malignancy in the oocytes. So we can basically start to use the oocytes collected for IVM as the fertility preservation procedure in these patients. And they have never ever had a chance to have fertility preservation before. So I assume that this is one group of patients where we can now start to use this perspective. So in conclusion, I think we are moving down the ladder, understanding the development of smaller follicles in humans, and this is paving the way to understand how we would develop IVF in the future. I do foresee that sometimes from now on, we would probably develop a exogenous free gonadotrophin infertility treatment, where we would just be able to do IVM collected from small antral follicles in the ovaries directly, and then avoid stimulation of the woman. We can do it in the 25 microliter droplet we are having in the lab. And this is, of course, a long-term perspective, but I do think that this is very interesting, and it is basically very encouraging for a research effort.